I'm Daniel Levine, and this is the Bio Report. Pancreatic cancer remains one of oncology's deadliest diagnoses, with standard treatments often offering only transient tumor shrinkage at the cost of grueling side effects and rapid resistance. Immuneering is using transcriptomic and informatics tools to design a MEK inhibitor dosed in intense daily pulses rather than continuously. This approach aims to restore more normal signaling rhythm in healthy cells while repeatedly ambushing tumors. Ben Zeskind, CEO of Immuneering, discusses how the company is using its informatics-driven dosing regimen to re-engineer targeted cancer therapy so it extends survival, delays resistance, and is better tolerated. Ben, thanks for joining us.
Thanks for having me, Daniel. It's a pleasure.
Well, we're going to talk about pancreatic cancer, Immuneering's novel dosing design, and its efforts to develop a treatment for this and other cancers using this approach. Well, let's start with the challenges of resistance and toxicity with targeted therapies today. Can you explain the problem?
Yeah, absolutely. So our goal is to keep patients alive and to enhance their quality of life. And the two things that you mentioned really get in the way of that with traditional targeted therapies. And if you look at how cancer therapies have traditionally been developed, they're developed to shrink tumors quickly, but it's not necessarily a durable effect. And that's why you see patients, why are patients still dying of cancer in 2026? There's a lot of treatments that can shrink tumors, but the effects aren't durable. Resistance develops, and the treatments are not generally well tolerated. There's generally a lot of fairly harsh side effects. I think we've all been conditioned to just kind of expect that cancer therapy is going to be grueling, and it doesn't have to be that way. When it's a design goal from the start, you can make a drug that helps keep cancer patients alive and is really well tolerated, and in fact can enhance the patient's quality of life. And so those are really the those were our goals from the start. From the time we started this company, we focused on overall survival, understanding the patients who were having long overall survival on existing cancer therapies, so we could figure out how to make that happen in more people. And that led us to focus on these fairly unusual priorities. How do you counteract resistance, right? How do you prevent the tumor from adapting and getting around your therapy? How do you improve the tolerability? And then other priorities. How do you prevent kind of the muscle wasting that causes patients to lose weight? So we just kind of had this different set of priorities from the start that were all focused around this goal of keeping patients alive and really helping them thrive.
Those goals led you to what's known as deep cyclic inhibition. In lay terms, what is that, and how does this address the problem?
Yeah, absolutely. So people say you have to outsmart cancer. Cancer's not actually that smart. It just has a couple of good tricks, right? And probably its one best trick that it has is that it adapts, right? It develops resistance. It finds a way around a treatment. And when you give most cancer therapies, they're given in such a way that they try to block the signaling pathway continuously, 24/7. So, all our cells in the body, our healthy cells, have signaling pathways that tell the cells to grow and divide. That's normal. That's healthy. Normal cells need to be able to grow and divide. But normally that signaling is transient. It's intermittent. Every now and then it says grow and divide, grow and divide, grow and divide. And what happens in cancer is that signaling pathway gets locked on. You have a mutation in a gene, often in that signaling pathway, which causes it to be locked on 24/7. So it's constantly shouting, grow and divide, grow and divide, grow and divide, grow and divide, nonstop. And so what most cancer therapies have done is they've tried to shut down that signaling pathway 24/7. They've tried to just silence it, but they can't really get it all the way off. So it ends up going continuously, but at a lower volume. So it's kind of like grow and divide, grow and divide, grow and divide, grow and divide, grow and divide. And that's not really restoring the normal healthy cadence of the signaling. Whereas what we do with deep cyclic inhibition is we're actually able to shut down the pathway more completely for several hours a day, and then we release it and we give it back. And that has two really important effects. When it comes to tolerability, we're essentially restoring that normal healthy cadence of signaling. So we're taking the pathway back to grow and divide, grow and divide, grow and divide. So the healthy cells are getting the intermittent signaling that they need to grow and divide. So you don't have the same kind of side effects that you normally would when you just shut down the signaling for the healthy cells. And at the same time, the cancer cells are deprived of that sustained high level of signaling, that grow and divide, grow and divide, grow and divide that they really need. So we're kind of depriving the cancer cells of that. We're giving the healthy cells what they need. And that's where you get the tolerability from the deep cyclic inhibition. The beauty of it is you're also counteracting the cancer's ability to adapt, because when you give it a nice, steady, predictable signal, you're kind of making it easy for the cancer to work around it. You're kind of it just kind of knows what it has to do. Whereas with deep cyclic inhibition, again, we hit it with this really strong pulse of inhibition of that signaling pathway. But then when we release and we give the pathway back, the cancer lets its guard down. It goes right back to using the same signaling pathway that it was before. And then the next day we hit it again just as hard, and we can play the same trick on the tumor every day, and it kind of doesn't learn. So by having this deep cyclic approach, we cause the tumor to let its guard down. We hit it again, and we're just not driving resistance in the same way. So as we talked about before, it's really this resistance and this tolerability that's prevented cancer therapies from helping patients live longer and really enhance their quality of life. And with deep cyclic inhibition, we've shown in our clinical studies that we can really do both of those things.
Your platform technology is built around finding tumors that are driven by overactivation of the MAP kinase or MAPK pathway. Can you give a sense of how your platform works? What are the core inputs and what are the outputs?
The way we really identified deep cyclic inhibition was through our informatics platform, and this is really about analyzing the gene expression signaling that's happening in cells. And what we were able to identify was essentially this concept I was talking about earlier. When we looked at existing therapies and how they impacted the cancer cells, we found that the early time points, the first few hours, they were really having a very good effect. They were counteracting the disease-associated signals. But by the time we got to 24 hours, they were actually making things worse. So that really our platform and the informatics aspect of it enabled us to identify the timing, to get that timing right of how long you want to shut down the pathway for and when you want to release it. And that's really what enabled us to develop deep cyclic inhibitors to really have the right timing. So we're we're preventing the tumor's ability to develop resistance. We're hitting the tumor hard, but we're giving the healthy cells back the signaling they need, so that there's a lot less in the way of side effects.
Many companies are going after RAS and the broader MAPK pathway. What distinguishes Immuneering in developing therapies that provide deep cyclic inhibition? How do you get to those therapies with your platform? Is it a matter of screening, or can you engineer those qualities directly?
Yeah, absolutely. You're right. I mean, the MAP kinase pathway drives half of all tumors. So this is a pathway, a signaling pathway that's telling a lot of different tumors to grow and divide. So there's a lot of people working on it in different parts of the pathway, but all the other approaches again are this continuous inhibition. They're shutting down the pathway 24/7. And the challenge with that again is that the cancer finds a way around. So when you shut down the pathway 24/7, the tumor develops resistance. It mutates again, and it finds a way to get around the treatment. Whereas with deep cyclic inhibition, we're able to achieve much better durability because again we're kind of keeping the tumor off balance, right? It doesn't have the pathway, then it has it again, then it doesn't have it, so it just can't it can't develop the resistance as effectively as it normally does. We're sort of counteracting its best trick, and then by having the tolerability that we have, we're enabling patients to stay on treatment longer. We're enabling them to have many fewer side effects, so we can combine more readily with others. So it's really the deep cyclic inhibition mechanism by counteracting resistance, by improving the tolerability, that just enables us to keep patients alive and enhance their quality of life.
Why did you specifically focus on MEC as the control point rather than looking upstream or downstream of that?
Yeah, so MEC is the best place to target this pathway because it blocks the widest range of potential mutations. Again, it's all about preventing the cancer from developing resistance, preventing the cancer from adapting, preventing it from getting around your therapy. And this MAP kinase pathway that drives half of all tumors, the signaling, it starts with a protein called RAS, then it goes to RAF, then it goes to MEC, then it goes to ERK. And so MEC is further downstream than RAS. Now, everyone, a lot of people are focused on RAS and inhibiting RAS. Sometimes you hear KRAS in particular. But the challenge there again is cancer's ability to adapt. So a lot of times when you inhibit RAS with a steady, continuous signal, a steady, continuous inhibition, as everyone else does, the tumor finds a way around, whether it's just amplifying RAS, making more copies of the mutant KRAS to kind of overwhelm the inhibition, whether it's getting a mutation further downstream in RAF to get around it. Cancer adapts, it develops resistance, it has these mechanisms. And by going further down the pathway to the level of MEC, we're able to block a much broader range of potential mutations. So this is all to the same goal of preventing resistance, blocking the tumor's ability to adapt, so that you can keep the patients alive much longer. And so MEC is an ideal target in that sense. I think people have long appreciated that MEC is a good target. It's just that it's generally when people in sort of prior generations of therapies have inhibited it continuously, they get a lot of toxicity. So here again, the deep cyclic inhibition not only reduces the resistance, but it improves the tolerability, and it just sort of enables us to really realize the full potential of MEC as a target, which is we think optimal because it's further downstream in the pathway. So it really enables you to get around resistance.
Your lead experimental therapy is in development for a number of cancers, including melanoma and non-small cell lung cancer, but its lead indication is in pancreatic cancer. What makes pancreatic cancer the right entry point for your approach?
Oh yeah. Well, look, I mean, pancreatic cancer it's a terrible cancer. It's a huge unmet need. Sadly, there's over 50,000 deaths a year in the U.S. alone. So there's just a real need for better treatments for this therapy. It's also the vast majority of pancreatic cancers, about 97% of pancreatic cancers, are driven by the MAP kinase pathway. So, given that we're targeting this particular signaling pathway, pancreatic cancer is a good place to start, not only because of the unmet need, but because the vast majority of pancreatic cancers are driven by this pathway. And what that means is we don't have to do any genetic testing before patients enroll in our trials. They can just you know, uh, there's no enrollment criteria around the particular mutations that they have, and that lets patients get on studies faster, right? If they have to wait and get tested for different mutations before they can join a study, it can really slow things down, and they obviously want to get treatment quickly. So, by starting with pancreatic cancer, there's sort of no genetic testing that's required. Also because our goal from the start has been to enhance survival. Sadly, patients on standard of care in pancreatic cancer, the survival drops off pretty quickly with time, and so it lets us uh, really show a pretty dramatic separation. So, just at the beginning of the month, we announced 64% overall survival at 12 months in first-line pancreatic cancer patients treated with our drug candidate atebimetinib in combination with chemotherapy. That's nearly double the overall survival at 12 months with standard of care gemcitabine nab-paclitaxel chemotherapy, which is only 35%. So 35% survival in the pivotal study of that standard of care benchmark, uh, compared to what we're seeing, which is 64% overall survival, as we reported earlier in January. So it just you really quickly see those really substantial changes in survival. And again, that was our goal from the start. That's why we prioritize counteracting resistance, improving tolerability, and it's great to see that kind of outcome in the data that we shared earlier in the month.
What did it take using your platform to get your lead candidate?
Yeah, I mean, again, we used the platform to figure out the timing, right? How do we design a drug with the right dynamics, right? What's the right amount of time to shut down the pathway, and then what's the right amount of time to give the pathway back, so that you can basically optimize. You can kind of think like an engineer, right? Immuneering is our company name. Think like an engineer, and you can really we used the informatics and the transcriptomic data to really look at the timing of what's the optimal characteristics of the deep cyclic inhibition, so that you're maximizing, really enhancing the patient's quality of life by getting rid of as many of the side effects as you can, and then really helping them live as long as possible by by counteracting the tumor's ability to adapt. And all that we used our platform to really design the molecule. And look, tebimetinib is novel composition of matter. We just had the U.S. composition of matter patent granted over the summer, and we developed it in house. It's not something we licensed in from somewhere else. We developed it using our platform, and that's why it's so exciting to see the kind of survival that we reported earlier in the month.
And to what extent do you view this as a proof of concept of the platform?
Oh, 100%. I mean, we have a pipeline of deep cyclic inhibitors preclinically that target other targets and other pathways in oncology. And I think I mean, look, in our phase 2A study in combination with chemo, there are only two categories of adverse events that we see at the grade 3 level in more than 10% of the patients. Two categories of side effects at that sort of high level, and they're both associated with the chemotherapy that we're combining with, and neither of them are seen when we treat with atebimetinib alone. So really, it's not clear that atebimetinib is adding much, if anything, in the way of harsh side effects on top of what you would expect with the chemotherapy alone. So I think that really validates that deep cyclic inhibition is able to drastically reduce the side effects compared to a continuous inhibition approach. And the survival that we announced at the beginning of the month, 64% overall survival at 12 months, nearly double the benchmark for standard of care. I mean, I think that really shows that our efforts with deep cyclic inhibition to counteract resistance, to help patients live longer is, I think is really being validated by the clinical results that we're seeing.
Well, can you go a little deeper on the clinical results beyond what you've said? What's known about the safety and efficacy of it?
Yeah, it's really exciting, and particularly, I mean, look, overall survival is the gold standard, right? I mean, this is what cancer drugs are approved on. It's the sole primary endpoint in our upcoming phase 3 study. So overall survival is the most important metric, and I think what's really exciting is that the separation from the standard of care benchmark has really held very, very consistently over time. So we announced 6-month data over the summer, 94% overall survival, whereas the benchmark for standard care is 67%. So that's a 27-point separation from standard of care. So really exciting to see that. And then at 9 months, we see 83% overall survival, whereas the benchmark is 47%. So that's a 36-point advantage. And then at 12 months, as I mentioned, 64% overall survival, whereas the benchmark is 35%. So that's a 29-point separation. So the fact that we're seeing such a consistent separation, from what's happening with patients in our studies versus the benchmark from the pivotal study of standard of care, I think that just adds a lot of robustness to the result that we're seeing. It's 34 patients, so it's enough to really say something. We have 13.4 months of median follow-up time, so really a good amount of time to see that. But what's really remarkable is it's not just the numbers and the survival. It's what we're hearing from the investigators who are treating patients in our study. So, on our call in January, we had Dr. Meredith Pelster, great oncologist from the Sarah Cannon Research Institute, and she described one of her patients in our study of atebimetinib plus chemotherapy. This is a first-line pancreatic cancer patient, and this patient had two lesions, both in the liver, that were rendered completely undetectable, just gone with the treatment. As the data cutoff, patient continues on treatment. It's been about 5 months. What's remarkable is this patient has seen dramatically improved quality of life. So at the beginning of the study, the patient had so much pain and weakness, and Dr. Pelster describes this on our call. So much pain and weakness, she needed family members just to bring her to the appointment. So it's a female patient. After being treated in our study with atebimetinib and the chemo, this patient regained the ability to drive independently, which is remarkable. The patient had been participating in a walking group in her neighborhood, had to stop due to the cancer. On our trial, was able to resume that activity, rejoin the walking group. So this is what we mean when we talk about enhanced quality of life, right? It's not just the absence of side effects. It's enabling patients to be able to go back to their activities that they enjoyed, to live their lives, not being restrained in the same way by the cancer. And pancreatic cancer patients frequently lose a lot of weight. It's muscle loss. It leads to weakness. This patient gained a substantial amount of weight, much healthier appearance, and all this Dr. Pelster talks about on the call. So it's really been remarkable to see that.
And are you looking at this both as a monotherapy and as a combination with chemo?
We are. I think most therapies are ultimately used in oncology in combinations because, again, by combining multiple therapies, you can block more of cancer's ability to adapt, right? More of the resistance mechanisms. The broader you can block, kind of the more the harder you make it for the cancer to adapt. So if you think of the MAP kinase pathway as a kind of a superhighway, cancer superhighway, we're kind of blocking all the lanes of the highway with atebimetinib. So we're making it really hard for the cancer to get around, but it could still sort of pull off onto a dirt road or a side road. And when we combine, it kind of puts up walls, if you will, on either side of the road. So it blocks some of those other pathways that the tumor could use. So I think combinations are really where it's at in cancer therapy. But the challenge is most cancer drugs are just so poorly tolerated that it's challenging to combine them. Tolerability is very frequently kind of the limiting factor to combinations, and because from the start one of our key goals was to enhance tolerability, enhance the patient's quality of life, because the deep cyclic inhibition enables us to really have a lot less of an effect on the healthy cells to really just maintain their normal signaling. We have this incredible tolerability profile where we can really combine really readily, and I think that's a strength of atebimetinib. So combinations really leverage that strength in terms of the tolerability and really help us maximize the durability, help patients live as long as possible by just making it harder and harder for the tumor to adapt and develop resistance and get around the treatments.
As I understand it, survival appeared to stay high as follow-up was extended rather than flattening out early, as occurs in many pancreatic studies. How has that affected your thinking?
We're just thrilled for these patients. I mean, to see the survival be nearly double what you would expect with the standard of care benchmark alone for these patients. I mean, and this is pancreatic cancer, right? I mean, this is not some indolent, slow moving malignancy, right? This is pancreatic cancer, and to have nearly double the survival. I mean, that means, think about it, for a patient with pancreatic cancer, first line pancreatic cancer, on the based on the benchmarks for standard of care, gemcitabine nab-paclitaxel, their chance of being alive after a year is about 1 in 3. In our study with atebimetinib plus the chemo, their chance of being alive at a year is almost 2 in 3. So that's just a remarkable difference, and to be able to do that with great tolerability, with patients experiencing the enhanced quality of life, like I described from Dr. Pelster's case study, it's just really, for us, it's thrilling to see that the goals we set out with at the beginning, to keep patients alive longer and enhance their quality of life, are really playing out the way we design it. And I think it validates the platform and the ability of deep cyclic inhibition to really achieve these goals.
How is what you've learned from your lead therapy fed back into the platform to improve the next generation of drugs?
Yeah, it's a great question. So, eh, the specifics of how we design a molecule to be a deep cyclic inhibitor, over the course of developing atebimetinib, we really learned a lot about how to do that. Right, and we refined our informatics and other aspects of our platform, and we're now applying all those lessons to kind of to our pipeline, to the preclinical deep cyclic inhibitors that we're developing against other targets and other pathways in cancer. So, I think the success and the validation that we're seeing in the clinic with atebimetinib has really helped us fine tune the approach to make sort of a whole pipeline of deep cyclic inhibitors that we're really excited about.
And how do you think about potential combination therapies? And does the deep cyclic inhibition enable things that a traditional MEK inhibitor might not?
Absolutely. Again, because the tolerability is so good, it really atebimetinib lends itself to many different combinations. And the first one that we're taking forward, beyond the pancreatic cancer we talked about, is a combination with an immunotherapy, an anti-PD-1 immunotherapy for lung cancer. And that's through an agreement with Regeneron. We're combining with their anti-PD-1 Libtayo. We expect to dose the first patient in that study in the second half of this year. And preclinically, we see really nice synergy between deep cyclic inhibitors and and immunotherapies like anti-PD-1s. And it's not just in our hands. There's actually a great paper by one of the leaders in the immunotherapy field, Jed Walchuk, where he specifically showed preclinically that not just MEK inhibition, but pulsatile MEK inhibition specifically enhances immunotherapy in a lung cancer model. So it's not enough to just inhibit MEK; it has to be pulsatile, and of course, deep cyclic inhibition is sort of the optimized pulsatile approach. So there's great data to support combining with immunotherapy. We're excited for that study. And then, we've shown preclinical data, with synergy combining with RAF inhibitors, with mutation-specific KRAS inhibitors. And so there's just a really wide range of combination opportunities that are enabled by, having a MEK inhibitor like atebimetinib that's so well tolerated and so durable, thanks to the way deep cyclic inhibition blocks, really mitigates the resistance mechanisms that the tumor would normally have.
How do you prioritize additional indications for atebimetinib, and how do you think about building your pipeline?
Yeah, it's a good question because there's so many different cancers that are driven by the MAP kinase pathway. So, as I mentioned, pancreatic cancer, of course, is first and foremost. That's where we're seeing nearly double the survival at 12 months in first-line patients in our phase 2A study, the data that we announced earlier in the month. And so obviously, that's our top priority, right? We're moving that to a phase 3. We've guided to dosing the first patient in the phase 3 mid-year this year. So we're excited about that. And the second priority, of course, is lung cancer. We talked about the combination study with with the anti-PD-1. But beyond that, look, colorectal cancer. There's a lot of MAP kinase-driven tumors, melanoma, AML, breast cancer. There's really a wide range of opportunities that we intend to pursue over time, because again, this target is so broadly important in cancer, and the deep cyclic inhibition approach enables us to to address it in such a tolerable way and with such durability that we think there's a really broad range of opportunities that we're excited about.
Where does partnering fit into your vision for the company? How do you decide what to keep fully in-house versus where to bring partners in?
Yeah, for us, it's all about maximizing the impact of atebimetinib, right? So, we're fully funded to run the phase 3 ourselves. So we're moving full speed ahead with that. As I said, we intend to dose the first patient by mid-year. So the phase 3 in pancreatic cancer is that's full speed ahead. We talked about the lung cancer study in combination with the anti-PD-1. And that was through an agreement with Regeneron. We've partnered in a lot of different ways. We were honored to have Sanofi as an investor in our financing in September. So that's another example, and we really look for opportunities where partnering can help us bring atebimetinib to more patients faster, whether it's new geographies, new combinations, or other kinds of opportunities. So that's really the metric when we think about partnering. But we certainly we maintain active dialogues with quite a few potential partners.
You mentioned the investment from Sanofi. They invested $25 million in a private placement concurrent with a $175 million public offering in September. How far will existing cash take you?
Yeah, so that all gives us cash runway into 2029. So nice runway, and we've guided to a top-line readout from our phase 3 in mid-2028. So it gives us cash runway beyond that top-line readout of the phase 3. So certainly very happy with that. And given the exciting data we're seeing in the phase 2 with 64% overall survival at 12 months, nearly double the benchmark, we're really, really excited for the phase 3.
There has been talk about the sector emerging from what's been called a biotech winter. You obviously had a successful fundraise in September, but I'm wondering if you've seen a change in attitude among investors and the types of questions or concern they express.
Yeah, look, I mean, the sector the sector is cyclical, right? It always has been, right? I mean, we started this company in 2008. And at that time, when you tried to pitch to investors, it kind of like, where'd everybody go, But we were focused from the beginning on this goal of keeping patients alive and helping enhance their quality of life. So I think we've persevered through ups and downs in the biotech market. And, I think we're, thrilled to have a great group of investors who enabled us to raise the funds we need to really take this drug all the way through phase 3 and beyond that top line readout. So I mean, the challenging biotech markets definitely come and go, but we've been resilient and cash efficient, and I think just persevered with this this really unique goal of keeping patients alive and enhancing their quality of life. And the data we're seeing for atebimetinib for survival and tolerability is really, really unique, and we're excited to move it forward.
Ben Zeskind, co-founder and CEO of Immuneering. Ben, thanks so much for your time today.
Thanks for having me, Daniel.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.